Let's go ahead and get started. I have quite a few announcements this morning. First one is to continue to plug the Advanced Molecular and Cell Biology Research Lab, Mutant Viruses from Hell. There's still a couple of spaces left in the Tuesday, Thursday afternoon lab. There is no particular prerequisite, or at least you don't have to take the recombinant DNA techniques lecture at the same time. We do expect you to have an advanced lab, i.e. a microbiology lab or a cell biology lab for that. The Virus Structure and Function Literature Review course, we have given preference to people who've signed up for the lab, but we're going to be opening it up to other people on Friday. So if you're still interested in this one-credit class, basically if you've taken molecular biology and or biochemistry, you should be well prepared for this. Another thing to announce, sorry it's a little hard to see his picture here. I've told you about this already, but now it's actually officially live. The Lester Newman Undergraduate Research Scholars Program. This is going to provide $5,000 to a couple of really enthusiastic undergraduate researchers who want to work in a lab this summer. Please apply appropriately. You should see this floating around on campus, also on the biology department webpage, and I posted it on my Facebook and Twitter accounts. Last thing is the department seminar tomorrow. Dr. Scott Freeman from the University of Washington has really sort of revolutionized a lot of the ways people teach undergraduate biology courses, and I've learned a few things from the things that I've heard about that he does before, but I'm really looking forward to his talk tomorrow. That will be at noon in, remember correctly, normal seminar room, yes, 247 SRTC, right across the hall from the biology department office. Should be a lot of fun. Apparently he lectures to the equivalent of principles of biology with 2,000 students in his class. And still gets interaction and how students are helping out with that. So I'm really interested to hear what he's going to say about how all of that is working. Friday we will have guest lecture, again, pre-Dr. George Kaysen, who's a graduate student in my lab, is going to talk about small RNAs and how those small RNAs have been used now for genome engineering, but also just in general small RNAs. And it's also completely unfair of me to ask him to do this because, as I think I've mentioned multiple times, you could do a whole course on RNA and small RNAs. And in fact, Dr. Lehman in the chemistry department is going to be offering that course next term. But before that, I wanted to finish up with transcriptional regulation, and particularly transcriptional regulation as it applies to regulatory circuits that we just briefly touched on at the end of last lecture. Think about positive feedback loops. We've talked a lot about positive feedback loops already, so this should be kind of review. But then also think about negative feedback loops and how all of these things start to tie together to give you some of the regulation that we see in cells, and also apply some of the tools that people have learned actually in electronics, among a lot of other things, in terms of thinking about understanding how biology works. And so I think there's a great cross-disciplinary way of thinking about biology from these points of view. And then we'll talk a little bit about epigenetics and DNA methylation before we move on to talk about some post-transcriptional regulation and just very briefly what we do there. And then we've got next week where we talk about methods, and then it's the final already. Where did this quarter go? Good question. I'm still not sure myself. So positive feedback loops. This is basically sort of the example we talked about last time with the genetic switch in lambda. You have the lambda repressor, which, quote-unquote repressor, because it's actually stimulating transcription of its own gene, and also the CRO protein, which is stimulating expression of the CRO protein. And so anytime you have this positive feedback loop, if you have a protein which is expressing its own gene, even after you have cell division, you can have, get that pointer to work, here we go. If you have this protein which is stimulating production of its own gene, even after cell division, And continually with cell division, as long as this protein is present, it will continue to stimulate expression of that same protein. So basically, you can also think about this as a cellular memory. There was some kind of signal that happened here, and very often a brief signal. Once you've turned something on, it stays on. And this is also very important from a development point of view. If you think about starting out some trajectory which is going to develop into liver cells or something else along those lines. So positive feedback loops are very common in biology. We already talked about them from heterochromatin and things earlier on. Positive feedback loops are, however, by no means the only kind of regulatory circuits that we have. We also have negative feedback loops. And so negative feedback loop is, instead of having an activator binding to its own promoter, now you have a repressor which is binding to its own promoter. And what that does is it makes sure that the level of that particular protein stays pretty constant, because the more you make of it, the less you're going to be transcribing that particular gene. So it keeps it at pretty much a nice equilibrium. Now, that's if you're regulating your own promoter. If, however, a negative regulator is regulating the promoter of a gene that makes a protein that negatively regulates the first promoter, what you end up with is this oscillation. And we'll see an oscillation step in just a second here, where you have expression of one protein until you get enough of that to suppress the first protein, which will then last for a while, and then you end up with suppression. So multiple rounds of suppression. And this is because it takes time from transcription, translation, DNA binding. And so you always have a time lag which happens between these things here. And then there's another system, and these are only a few of the various different circuits. And again, you can learn a lot from thinking about electronic circuits from the same point of view here. Feed-forward circuits. Here we have an activator which activates a second activator gene, and then both of them together have to activate a third gene. And this is what we call a noise filter. And then we can look at this in a little bit more detail on the next slide here. So again, just a quick reminder, positive feedback, this is keeping one state, remembering what's happened in the past. So positive feedback loop, turn something on, it stays on for longer periods thereafter. Negative feedback, keeping levels constant, because too much of something, you're not making any more, not enough of it, you're still going to make a little bit more of that. Then we have the negative, just a second, excuse me, the repressor repressing a repressor, and that gives you oscillations, and we'll look at an example for that on the next slide. And then we've got this feed forward. Yes, your question. Yeah, so the repressor is repressing the next repressor. Again, we'll see that this example on the next slide, so we'll take a look at that. I should have maybe switched these up. But yeah, the idea of a feed-forward loop, and this is two positive activators that have to act on a downstream gene, is what this does is if you have a very short input pulse, again, which is, here we have our pointer, a short input pulse, what this is going to do is it'll turn on A, but there won't be enough time to turn on B as well before A is turned off again. However, if you have your input long enough that A can turn on B and you still have A around, then you end up with your final output. And so just the fact that you've got these two regulators, one that's regulating the next one, means that you have a way of making sure that you have a long-term signal, and basically it's a noise filter. If you have a little bit of noise like this, small variations, nothing happens because you haven't had enough time. However, if you have a longer one, then you will have this output. And this turns out to be very, very useful if you think about hormones, for instance. Small amounts of hormone that would be produced under some conditions. Are you going to want to have a response to it or not? If, however, you have a long period of hormone production, then yeah, we better turn something on. Yeah? Okay, so the question here is this: how does this relate? Now, I'll paraphrase the question and correct me if I'm wrong again. Does this relate to the heterodimer formation or dimer formation giving you more of a on-off switch? I would say that in this case, that would be one of these steps. So one of these activators together would have that. So the idea is each of those individual switches, and when I say a switch there, it means you're turning on a particular gene at a particular concentration of your regulator in this case. And so this is more of a concentration-dependent issue. This is sort of the next step where you will now have the activation of one particular gene, which would then lead to the activation of a second gene in this kind of feed-forward process. So that's sort of the first step before that second step. And this is a bigger picture kind of thing. The dimerization is a smaller picture. So those fit together. And then, as promised, this feedback loop of now negative regulators, so the positive regulators are leading to this filter. The feedback loop of these negative regulators gives you oscillations. And so here we have gene A at the top, which makes repressor A, which will repress production of repressor B, which will repress production of repressor C, which will repress production of repressor A. This was predicted, theoretically, and then people actually did this. They made these particular constructs and put them into cells, and they saw exactly this process down here at the bottom. And so this is the prediction. If you have numbers of proteins over time, if you make this repressor, make enough of it, you'll shut down B. Make enough of B, you'll shut down C. Make enough of C, you'll shut down A. But if you don't have these repressors, what happens? You have expression of all of these genes. So in the absence of repressor, you make everything. And then as you make more and more, you shut these things down. Once, however, these proteins have a limited half-life, and in this particular case, it's a very limited half-life, then you'll see this oscillation. Why do we care about oscillations? What's that big yellow thing in the sky? I know we don't see it all the time in Portland, but these are very important for circadian rhythms. So all of our cells are timed to do things at different times, and that timing gets reset by the sun. But basically, even if you keep us or other animals in the dark at about a 24-hour cycle, you will see genes get turned on and turned off, sleep, wake, etc. So this kind of process is exactly what happens in normal circadian rhythms, is these repressors following along with each other. So this is that big picture kind of thing. Well, we've got positive feedback loops, negative feedback loops, together two positives, together two, or in fact matter, more negatives, leading to these kinds of circuits. And it's a whole combination of these different circuits to look at regulation of various different things. And they've got this ridiculously complex image in the textbook for a very simple process happening in development. And people are now trying to piece apart how all of these things are working. And can you make predictions in terms of what genes are going to get turned on and turned off, knowing what you do about the individual components? And so that's a big open area. A lot of people call this systems biology. It's understanding that system and then trying to use what you can understand there to design organisms or pathways, et cetera, which turn out to be incredibly useful. So questions on this aspect? Yeah? Okay, so the question here is, why are we looking at fluorescence here as opposed to, we're just talking about repressors here, right? So this particular experiment, again, that was done in cells, had a readout or a reporter gene which was fluorescent. And so you're looking at the expression of that reporter gene, which is being regulated by all of these repressors. Yeah, and so the idea here is basically what we're looking at, and so this was the theoretical progression here. Now if we're just looking at this one particular gene, so this is the one which is regulated due to the gene regulatory sequence right here, this would correspond to those red peaks. So you see it's increasing here, and in theory at some point it should level off. I think that they did see that it leveled off later on. I haven't gone back and looked at that experiment. But yeah, it's the idea. This is sort of the very beginning of that process. Okay, so I want to switch gears a little bit and talk about some weird things that happen in mammals. Why do we care about mammals? Because we are mammals. And this has to do with the fact that we have, or slightly more than half of us, have 2 copies of the X chromosome, and less than half of us have only 1. So the vast, well, not the vast majority, the majority of people have 2 of these, but you only need 1, with some very slight exceptions. Again, this is biology. And so if you have 2 X chromosomes, and they're actually being expressed actively, this is a big problem. And in fact, cells will die and embryos will die if the 2 X chromosomes are being completely expressed. And so one of them gets inactivated. And this process is called dosage compensation. Been very well studied over the years, trying to figure out why 1 and only 1 of the X chromosomes in mammals. And it turns out there's dosage compensation that happens in all kinds of different organisms as well, not just in mammals. But as far as we're concerned, we're just going to look at dosage compensation in mammals, which has to do with the ratio of the X chromosomes to autosomes. And so sort of how this is measured. We're not going to get into how it's measured. If you're interested, you can email me. I can send you the 60-page articles on how people figured this out. But basically, if you have an X to autosome ratio, which says you're female, you've got 2 X chromosomes, then this turns on a pathway which will inactivate 1 of those X chromosomes. But this happens only after about 1,000 cells have been made in an embryo. So the first 1,000, well, actually, back calculate 1,000, that'd be 1,024. How many cell divisions do you get? I think like 8 cell divisions would have to happen before you had 1,024 cells, if you could hang on to all of them. But at that point, then 1 of the X chromosomes get inactivated. And it seems to be really random which one of those gets inactivated. And once, however, that happens, you have a positive feedback loop, which will lead to the rest of that chromosome being inactivated, and that inactivated chromosome being maintained over time, and all of these cell divisions. So what is that inactivation of the chromosome? Not surprisingly, it's heterochromatin, which means that it's been completely compacted. And in fact, you can see this heterochromatin when you look at female mammalian cells, because there's a really, really compacted chromosome, also known as a bar body. In fact, for a long time, this was how people decided in the Olympics whether you were female or male, was if you had a bar body or not, which turned out to be an interesting problem that, again, we can talk about offline at some point. But this heterochromatin is different than some of the heterochromatin we've talked about before. Heterochromatin that you have at telomeres and you have at centromeres is heterochromatin all the time. But here, this is heterochromatin that is sometimes heterochromatin, and particularly in some cells, and in other cells, it's not. So this we call it facultative heterochromatin. So there's constitutive heterochromatin and facultative heterochromatin. Facultative is reversible. It's only reversible when you go back and actually make your gametes. Or if you talk about heterochromatin in terms of gene activation, that's going to be going from heterochromatin to euchromatin. But there are parts which are constitutive heterochromatin, and again, those are telomeres and centromeres. They're always compacted at all the time. One nice example of this randomness, and you can tell that there's random inactivation of paternal and maternal X chromosomes. Calico cats, you never see a male calico cat because they're not mixed, because they only have one X chromosome. Whereas female calico cats can have lots of different colors, and that's because those coloration genes are on the X chromosome, and some of those will be inactivated here, some of those have been activated there, and basically it means that all females are mosaics in terms of expression of X chromosomes. Now, how do we get one of those X chromosomes inactivated? And this is a nice lead-in to the lecture which George will be giving on Friday. All has to do with RNA. So there's a specific part of the X chromosome called the X inactivation center. It's a part of the genome where you can have expression of a particular RNA. People look for proteins literally for years to try and show that this is what was causing the inactivation of one of these chromosomes, and then they found out actually it wasn't the proteins at all, it was the RNA. So this X inactivation center, so XIST RNA, is made through regular transcription, RNA polymerase II, from this particular region. But then what it does is it base pairs with this region, and then will base pair with itself, and base pair with itself, and base pair with itself, in order to end up covering the X chromosome that this one part was expressed from. And again, it appears to be at random, but once you have this expression, due to the positive feedback loop, you continue to express from this particular chromosome. It will cover this chromosome in this RNA. And then you have proteins that associate with that will take acetyl groups off of histones. So again, leading to lack of expression of the various different genes. Also will methylate histones and put in alternative histones through histone chaperones. And again, all of these through interactions with this long non-coding RNA that, again, George will talk a lot more about on Friday. And then finally, you have methylation of the DNA. And it's the combination of all of these things: hypoacetylated histones, methylated histones, alternative histones, and DNA methylation that leads to this really amazing compaction that you can literally see in bar bodies in female mammalian cells, and this very, very compacted chromosome. Yeah? Mm-hmm. Yeah, so, okay, let's just repeat this. But yes, if you look at the cells which have derived from those first, you have about 1,000 cells, and then 500 of them are going to inactivate the paternal X chromosome, 500 are going to inactivate the maternal X chromosome. All the cells that are derived from each of those 500s are going to keep that particular case, but it seems to be pretty random. So the question is, does it provide a really big difference? If there are differences in the alleles in the X chromosomes from males and females, that can be a problem. But generally, most of the X chromosome is pretty similar from their maternal and paternal ones, so there doesn't seem to be a huge difference. And those alleles that turn out to be really negative have been selected out of the population because they don't create a viable organism. Yeah, in the back. So yeah, the question is basically, is it really random or is it not random? Maybe that's a better way of putting it. So the answer to that is I don't know. But as far as my understanding is, it actually seems to be more of a mixture. So at 1,000 cells, You've already differentiated and decided which direction some of those cells are going to go. And again, take developmental biology next term. I'm sure you'll learn a lot more about it. So I don't think that one particular organ is developed from one of those original, again, 500 cells, if we're taking 1,000, that would just end up being all liver. So I think that most organs are actually a mixture of the two. Right in the back, and I'll get back to you. Okay, so the question here is basically, as you have cell division, why do you have one that stays the same? And so it seems that once you have expression of that locus, that will stay being expressed over time, and probably because of when you have the pairing of the chromosomes at mitosis, and then those separate, that will actually be the one which is already there. The RNA seems to jump over to the other one and stay on that particular one. I'm not sure I completely understand your question, but basically at the 1,000 cell, before you get to 1,000 cells, you have 2 X chromosomes which are both being expressed. Then at about the 1,000 cell stage, half of them inactivate one of them and half of them inactivate the other. Individually in each cell, yes. And so that's when you have the signal, that turning on of the XIST happening. Yeah, down here. Okay. Okay, so I mentioned this DNA methylation at the end here. I want to spend a little bit more time talking about DNA methylation. It turns out that it's not just important for inactivation of excuse me, X chromosomes. True in almost all facultative heterochromatin. We talked about this a little bit when we talked about formation of heterochromatin. We talked about chromatin remodeling complexes and histone modifications earlier on. I want to focus in on what's actually happening here. So DNA methylation, when we're talking about it from this point of view, and again leading to heterochromatin formation, happens at the 5 position of cytosines. So methyl groups get added right here. This is where this is attached to the ribose. So this is the equivalent position to where you have a methyl group in thymine, but no change in base pairing. All your base pairing is happening over here on this side. So where do we see this kind of cytosine methylation taking place in cells? It seems to happen where you have a cytosine guanine right next to each other. And why is that? That's because there's a protein which will put methyl groups onto guanine residues when you have a CG on one strand and a GC on the other strand. So it's basically 2 GC base pairs, opposite orientation relative to each other. That gets recognized by a methyltransferase. And so a methyl group gets put onto the guanine, and that happens on one strand. And then there's something called the maintenance methyltransferase, and this is a lot like what we saw when we talked about bacterial regulation. This is now happening in eukaryotes. In eukaryotic systems, if you have this CG, which is methylated on one strand, so you have a heterodimer, the CG on the other strand will also be methylated through this protein called the maintenance methyltransferase. So replicating methylated DNA, this maintenance methyltransferase will make sure that your DNA is methylated on both strands, and this makes perfect sense if you think about it being important for heterochromatin. You want to have this heterochromatin formation. You want it to keep being heterochromatin. You keep that methylated group which is actually present there. So how does DNA methylation happen? It's DNA methylation, but also, and this is in fact what we looked at before, we have histone modifying enzymes that eventually lead to DNA methylation. This DNA methylation through the maintenance methyltransferase will stay methylated, but methylation by itself isn't enough. You also have to have methylated DNA binding proteins in order to get full compaction of the DNA in that particular position. And so it's this modification of the methyl groups which helps part of the process of getting your full heterochromatin formation. A lot of people also talk about this in terms of silencing. and hopefully some of you remember the silencing we talked about last time for silencing of the yeast mating type loci that are not in the mating type, and I posted a couple of pages from the previous textbook on how we know about silencing there. Part of what we know about silencing is that methyltransferases are important for yeast mating type silencing, also true for silencing of DNA in other eukaryotes as well. So this silencing process is, again, changes in histones, changes in the DNA, and binding of these methylation proteins. And it turns out that the process of going from euchromatin to heterochromatin, and again, going from active DNA to non-active DNA, is about 1,000,000-fold in eukaryotes. So euchromatin, you have 1,000,000 times more expression of a particular gene than you have in heterochromatin. And so that's partly why, when we talk about transcriptional regulation and getting things to the appropriate promoter, that's why moving all of these histones around, dealing with all of these issues, has to do with these chromatin remodeling complexes. So once you shut everything down, it's really, really, really turned off. In bacteria, you actually only see about 1,000-fold regulation. The really on genes are 1,000-fold more than the really off genes. And that's probably for a couple of different reasons. But one of the main ones, and we looked at the human genome, most, well, not quite most, but 40% of the human genome is made up of what? Transposons, retrotransposons. If retrotransposons were jumping around the genome all the time, what would happen to our genome? Get pretty shredded pretty quickly. So it turns out that a lot of this silencing seems to be important for shutting down the activity of transposons as well. And so if you have problems with DNA methylation, you have problems with heterochromatin formation, you see lots of genome instability because transposons start to move. So one thing that people noticed when they looked at these CG sequences, and again, CG on one strand, GC on the opposite strand, what they found is that there are very high concentrations of, when I say concentrations, just sequences. If you look at the human genome, lots of regions where you've got lots of CG, 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 CG. But it's underrepresented from what you would expect randomly throughout 6 billion base pairs of DNA. Now, why is that? What happens to pyrimidine residues, and particularly cytosine residues? Because we're in an aquatic environment at about 37 degrees Celsius. What happens? Deamination happens. What happens if you deaminate a 5-methylcytosine? Pardon? You don't activate it. What happens if you deaminate a 5-methylcytosine? You end up with a thymidine. Is thymidine something which is recognized by DNA repair glycosylases? No. So if you have methylation of cytosines over evolutionary time, you will actually have lost those cytosines and turned them into thymidines. So what this means is, if you have regions of the genome that have lots of GC, 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 it means they're probably never methylated, or very rarely methylated. Because, if you have methylation over, again, evolutionary time, you'll get rid of these things. And so what turns out to be really useful, methylation, again, is usually leading to heterochromatin formation, etc. Heterochromatin formation is giving you silencing of the particular genes which are there. Well, if you don't have methylation, what does that mean? That's a region which is going to be euchromatin most of the time and is usually going to be expressed. And it turns out you can literally go through the human genome and find these areas where you've got lots of GCs, and those match almost perfectly to gene regulatory regions of the protein coding genes, which is, again, a wonderful thing. And particularly, these are the ones for genes that are being expressed or transcribed all of the time. So ribosomal protein genes, for instance, down here, particular metabolic genes, other metabolic genes, very often have these people call CPG islands here because they've never been methylated over, again, evolutionary time scales. So really useful way of just literally looking at sequence and then being able to predict what you're going to find there. Turns out that methylation is not just used for turning on heterochromatin, and you can see that you have specific regions of the genome which get inactivated. There's also this process called genomic imprinting. This has to do with a slightly different way of expression from one chromosome versus another chromosome. And these happen in autosomes rather than in the sex chromosomes. And what this process is that one of the chromosomes, and in this case, the male chromosome, is methylated, and the maternal chromosome is unmethylated. And what that does is it means that you're always going to be expressing things from the maternal chromosome. I think that's getting back to your question at the back about which, under some conditions, are you getting just maternal and just paternal. This happens in some of those autosomes, but not in the X chromosome. So here, turns out that this is important for neural development, etc. This methylation is turned off when you have meiosis and gametogenesis in females, but is put on in all males. So if you think about a particular allele here, so this is going to be the mother, this is the father of the mother, the father of the father, which now have these modifications. That's going to be the red allele. The yellow allele here is from each of their respective mothers. When you do undergo gametogenesis, you separate the red allele from the yellow allele. In eggs, these do not have methylation. In the male, in the sperm, you now have methylation of both of these alleles. And the offspring of these two, even though at this point this methylated allele up here at the top was in mom and dad, the offspring, it's always going to be dad's alleles, and it could be two different alleles at that point. So here we have methylation of the yellow allele and methylation of the orange allele or the red allele. And what this means is, if you think about these different alleles and you're trying to do genetics, classic Mendelian genetics, you go, wait a minute, this doesn't make sense. And eventually we figured out it had to do with this methylation, and all through this whole process. There are about 100 genes in both mouse and humans that have this kind of inheritance pattern. Now, 100 protein coding genes versus 20,000, it's not very many of them, but a few of them do actually turn out to be very important. And one of the best studied of those is this protein called the insulin-like growth factor, insulin-like growth factor 2, actually, where we have methylation that takes place not in a promoter sequence, but in an insulator sequence. So, what the heck is an insulator sequence? What do insulators do? They isolate and protect promoters from what? From gene activator proteins that have bound to cis-regulatory elements. So if you have an insulator and you have to have an insulator binding protein, that will block the activity from a cis-regulatory element on a particular gene. If you have methylated DNA, that protein does not associate here. And so now this cis-regulatory element can actually turn on this IGF-2 gene. And this turns out is absolutely critical for fetal development in placental mammals. So without imprinting, you don't have this taking place. And it's not regulating a promoter, it's now regulating binding by an insulator binding protein. But binding, why is it changing binding? Well, if you think about a big methyl group sitting in the major group of DNA, which is what happens when you have 5-methylcytosine, then it's going to change the DNA binding protein association here. It doesn't change your base pairing interactions, but definitely is going to change what kind of proteins can interact with your DNA. Yeah? Yeah. Okay, so here, in this particular case, in IGF-2, there's methylated DNA. So you're methylating the DNA, which is that insulator sequence. The methylation of that DNA sequence doesn't allow the protein to bind there, which is going to give you your insulator activity. So it's blocking the binding. There's another case here where we have, and again, this is the shout out to George's lecture on Friday. You have methylation in different genes, and I can't even remember what this one is, so I'm not going to ask you to remember it either. But it's expression of a long non-coding RNA, and this long non-coding RNA is transcribed from a particular promoter. This promoter is imprinted or methylated in, I forget if it's males or females here, and then doesn't allow expression of this non-coding RNA. This non-coding RNA, as George will talk about on Friday, lots of these non-coding RNAs will bind to proteins, particularly histone-modifying proteins. These histone-modifying proteins will cause compaction of the DNA heterochromatin formation. So if you have this transcript for the long non-coding RNA, this particular gene is silenced. If that promoter is methylated, you don't have binding by the general transcription factors. You don't have binding by the RNA polymerase. You don't get transcription, so you make this gene and protein in the opposite orientation. So silencing here due to a promoter, and activation here due to methylation of insulator elements. So two different kinds of things that happen with imprinting. So now, zoom out a little bit. I wanted to talk a little bit about epigenetics. Probably all of you heard about epigenetics. Epigenetics is the latest, hottest field, or one of the latest, hottest things here. Oh, let's back up one. Skipped over. So the whole idea of epigenetics is inheritance, sort of from cell-to-cell division, getting back to the questions in the back about X chromosomes. And it's an epigenetic inheritance. If you think about that one cell of the 500 in the 1,000, which replicates and passes along that information to the next cell, but it's not a change in the genetic information. It's not a change in the DNA sequence. So epigenetic inheritance is just inheritance of some state of the cell as you have cell division, or for that matter, from one organism to the next organism. So we've already talked about this as being positive feedback loops. We have positive feedback loops here down at the bottom. You can also have DNA methylation, and again, through these maintenance methyltransferases. Once you're methylated, you stay methylated. And we've talked about histone modifications. Once you have histone modifications, and this would also be thinking about X chromosome inactivation, these guys are inherited with time. What we haven't talked about are these prion proteins, where the presence of one protein causes the folding to change of a different protein. This can also be maintained after cell division takes place. And these are sort of the four main kinds of epigenetic inheritance. So modification of DNA, modifications of proteins, and then presence of proteins inside cells. There are other kinds of epigenetic inheritance, but these are really the major ones in terms of looking at how you can inherit the state of a cell as opposed to just the genetic sequence in any particular cell. Yeah? Mm-hmm. Okay, I'm not sure I quite understand your question. Could you maybe restate it? So you got like different cell compartments, or because it's sort of from cell to cell, but you can also have this from organism to organism. So if you maintain any of these states, for instance, like imprinting from one generation to the next, that can actually sort of look at, and it turns out that people have looked at some of these diseases. For instance, if you have a epigenetic change in your grandmother, that may actually skip a generation, and then you see some kind of output later on. And so it can literally be from generation to generation. It's not just from cell to cell. We're here looking at cell to cell, but you can definitely have from parents to offspring. Oh, okay, so the question really has to do with prions and prion clusters. Again, this is sort of focused on the cell biology and the molecular biology. So in that case, yeah, there's some exceptions to everything, of course. This is biology. But we're not really going to get into that in too much detail. But the whole idea of, and I think this one better stated down here, we have these positive feedback loops. If you have the presence of a protein in a cell, be it a sperm cell or an egg cell, as long as that positive regulator is regulating itself, that will then lead to more changes which are happening. And the fact that these modifications, like DNA methylation and histone changes, these are in positive feedback loops, so they stay over generations. And that can be cellular generations or from organism to organism. And the idea here is that, getting back, as I didn't want to get too deep into this, but with prions, if you have one of the inherited prion diseases, those actually do seem to be able to be transferred as well, and that's that particular state. So if you flipped on the prion misfolding process, that seems to stay over generations. And again, it's a non-genetic change because it's that state of the protein which has changed as opposed to the genetic sequence. Yes, no, happy. I think I may have actually lost my clicker questions today. I thought I had some more, but oh well, I guess we somehow managed to lose them. Drat. Don't everybody leave. No. Doesn't give me a chance to have my drink halfway through. So this is transcriptional regulation. All done. Yay, happy. Good, no. Okay, let's move on and talk about post-transcriptional regulation. So now we have all of our transcripts started, and actually transcriptional regulation is really about transcriptional initiation regulation. So this is moving all the chromatin around, getting a pre-initiation complex, getting it actually transcribed. But in some cases, you start transcription and then the transcript stops. This is what's called attenuation. We'll look at that. Capping has to happen. Capping also can be regulated. Splicing gets regulated a whole bunch, and we'll talk about all of these. In some cases, we're finding actually more and more of this is happening. Once messenger RNAs are made, many of them get edited, and so this is, you can also think about it to some extent a epigenetic change, is because you modify an RNA, and sometimes that modification of the RNA can actually lead to a very different outcome in terms of translation. And then RNAs get exported from the nucleus. Again, this is another step where you can have regulation. Once the RNA gets outside of the nucleus, it can be put in specific places. We already talked about this when we talked about bicoid and hunchback. Those end up at particular places in the embryo. Where transcription starts or not, as the case may be, can be regulated. We already talked a little bit about recoding of translation, frame shifts, and selenocysteine incorporation. And then just the very amount of RNA that you have is going to finally determine how much protein you have at the end here. So just regulating transcriptional initiation is not enough if you want to try and understand where you have all of your proteins being expressed. So let's talk about a couple of these now briefly. One of them is really well understood in bacteria. There are few of these in eukaryotes, but mostly this is present in bacteria. These are what are called ribose switches. And a ribose switch is just a sequence of RNA that instead of coding for a protein can make a structure which can then change transcription. So in this particular case, we have a ribose switch, which is, again, an RNA sequence that forms a structure. If this structure binds to a particular molecule, in this case guanine, then this structure causes transcriptional termination to take place. And any time that you have premature transcriptional termination, that's called attenuation. These RNA structures, which will bind to things, are called aptamers, and it's literally an RNA sequence that forms a structure that has some kind of function. And again, take Dr. Lehman's class next term because he's going to talk a lot about all these various different aptamers. The thing I particularly like about this kind of regulation is it's all RNA. Has no proteins involved in the regulation here whatsoever. It's just that RNA sequence interacting with some kind of ligand, some kind of small molecule, which leads to transcription termination. Why do you think I've squiggled this out here? Because we're talking about bacteria. Bacteria don't have C-terminal domains on their polymerases. Alternative splicing. We've talked a bit about alternative splicing already. Just when we talked about splicing. Alternative splicing happens much more commonly in the larger eukaryotes than in the smaller eukaryotes. And that's what my yeast biologist friends taught me. They're not larger. They're not higher. They're just larger eukaryotes. And you can have basically both negative regulation and positive regulation, again, mostly having to do with these SR proteins that we talked about before. And what they do is they will block various splice sites if they're negative, or they'll stimulate splice sites. And in that process, you can have one gene here. This would be one primary transcript. This is going to lead to lots of different polypeptides. And we talked about this example before. This is that Drosophila gene that has over 30,000 different kinds of splicing that can take place in that gene. There's one particular example that I wanted to talk about. This has to do with dosage compensation, but not dosage compensation in mammals, but dosage compensation in fruit flies. This is basically leading to, again, male development or female development, all based on the ratio of X chromosomes to autosomes. There are 3 regulators here, 2 of which are splicing regulators, and then the last one is a transcriptional regulator. So in the upper case here, we have the sex lethal. So any given sex, it's a lethal mutation. That's how all of the Drosophila geneticists name their genes. So the sex-lethal gene in males is going to not be expressed, and you have a splice site that leads to a non-functional protein. So not expressed here. However, in females, sex-lethal is expressed, and again, I've got a 60-page paper that tells you all about how this works. But in females, sex-lethal is expressed, and it's a splicing suppressor which gives you a functional protein. So this functional sex-lethal protein has a positive feedback loop, which, again, once you're female, you stay female, that leads to blocking of this particular splicing here. That blocking of splicing, yes, it's a blocking of splicing, but it turns out to be a positive regulator, a positive feedback loop, gives you this functional protein. This functional protein will also block splicing in the transformer protein, and a nice thought experiment when you're thinking about reviewing this, think about why this is called the transformer protein or the transformer gene. So the functional transformer protein is now a splicing activator. This splicing activator will give you a double sex protein, and again, you can think about why it's called double sex a little bit later on, if the functional splice stimulator gives you a specific C-terminus of your protein that now will repress male genes, if you don't stimulate this weak splice site, now you have part of the protein which is going to repress female genes. So male repress female genes, female repress male genes. And this is a good indication about why these genes are called what they are. So I'll let you continue to think about that. Another one of the well-described understanding of post-transcriptional regulation has to do with tailing. So we had splicing and tailing. I haven't talked about capping. But different tails. How do you form poly A tails? What has to happen? You have to have cutting of your transcript. That cutting happens with CSTF and CPSF. Those have to bind to your RNA and cut there. Once you cut, then you have the poly A polymerase, etc. So in B cells, in immunology, B cells are those cells that produce antibodies. You can have two, this is again an oversimplification here, two different kinds of B cells. You can either have B cells that have antibodies that are bound to this B cell with membrane-bound antibodies, or B cells can be secreting antibodies. So secreting antibodies or binding to membranes has to do with the presence of this hydrophobic stretch of amino acids, which is only present if you have a longer piece of RNA that encodes this hydrophobic stretch of amino acids, or if this particular hydrophobic part of the protein is not made, then you have secreted antibodies. How do you get this? Turns out that there are different binding sites for CSTF. If you have low amounts of CSTF, then it will bind to the end of your primary transcript. You'll end up with a poly A tail here. On the other hand, if you have lots and lots of CSTF around, there's actually a weak binding site for CSTF. That's a long way away from the consensus sequence. It's in this first intron of this antibody gene. Now, intron, it shouldn't be there, right? It's all gone? Well, if you think about post-initiation messenger RNA processing, how is it happening? It's happening as the RNA is being made, because you've got all of those extra proteins sitting around in the C-terminal domain of RNA polymerase II. So as you're being transcribed, you basically have a competition between splicing, which would splice out this weak binding site for CSTF, where if you've got a lot of CSTF around, as you're transcribing, you're actually going to cut the messenger RNA right here, and you end up with a poly A tail and no hydrophobic region. So alternative tailing leads to secreted antibodies versus non-secreted antibodies. And these are individual examples, but hopefully you can think about other kinds of things that should be happening here. Again, important for the alternative splicing and for this alternative tailing, you have otherwise very similar proteins, just little changes which have happened to them. And usually at the C-terminal end of the protein, because you're going to be transcribing from 5' to 3'. Your translation goes from N to C-terminus. And it's going to be the C-terminus which will be different because of whatever changes have happened there. So we can change the RNA in that way. You can also change RNA based on RNA editing. And again, this is something that we're finding more and more happening in lots of different messenger RNAs, and again, the textbook is out of date on many of these. But the ones which are best understood are these so-called adenosine regulatory proteins, the adenine deamination. Adenine deamination particularly leads to inosine formation. This is what happens in mammalian systems. There are mitochondrial modifications that happen as well. I'm not going to talk about those, so they won't be on the exam. But this adenine to inosine, what is the really cool thing about inosine that we all remember from when we talked about translation? Can base pair with lots of different other bases. And so the change of adenine to inosine will give you different base pairing interactions that can happen, but now not from the tRNA side, which is where we talked about before. Now it's on the messenger RNA side. But it turns out the base pairs that it forms are exactly the same. So you end up with different amino acids at particular positions in your messenger RNA. Turns out that this happens because of the presence of secondary structures in your RNA that are then recognized by this sequence-specific RNA binding protein that also has this enzymatic activity that leads to adenine deamination. There also are lots of proteins that we'll talk about a lot more in virology next term, these so-called apobec proteins. These also heavily modify RNA and are very important for protection against viral infections, particularly for things like HIV. This is just what happens here. If you have editing in this particular protein, it's another editing site. This editing changes a CAA to a UAA. What is UAA? It's a stop codon that will change your protein, makes it a lot shorter, rather than a protein which is a lot longer. This will give you different activities of these different proteins in different parts of the cell. Now we're talking about HIV, leads me into the best understood regulation of RNA export. RNA export in HIV. HIV, as we talked about really briefly when we talked about retrotransposons. Retrotransposons, some of them are very similar to these retroviruses. They've got one big RNA, which is going to code for all the things you need for a viral genome, but it also makes smaller RNAs. So what has to happen for this virus to be functional is you have to get that whole RNA exported out of the nucleus. The way that works is there's a specific protein, the so-called Rev protein, that's made through splicing that happens in the RNA while this gene is being made. Once you've made this Rev protein, it gets imported back into the nucleus, and then we'll take the whole messenger RNA out into the cytoplasm where it can be made into a whole virus. The similar kinds of things happen with different messenger RNAs that are made inside the cell of different sizes, and this transport process, which is always an active process moving out of the cell. Then the last thing I want to talk about in terms of messenger RNA has to do with where messenger RNA is located inside the cell. Again, we already talked about this with bicoid and hunchback. The reason that you've got gradients of bicoid and hunchback from the anterior end of the embryo is because the messenger RNA that's made by the mother in making the egg for bicoid and hunchback is always put at the anterior of the embryo. And that messenger RNA is going to be translated at that point and will diffuse from that point through the rest of the embryo. Talk about translational control actually on Monday, because George is going to talk about small RNAs and some of these larger RNAs on Friday. See you then.